Our next phyla that we're going to be discussing are the cnidarians. Now, if you thought that C was supposed to be pronounced, it's actually not. It's actually silent. And so the correct way to pronounce this phyla is Cnidaria. And cnidarians have two specific body forms. Not all creatures have both forms. Some of them have just one, some of them have just the other, some of them have both. But the two forms are a polyp form, which is going to be a stationary form. It's not going to move around. And a medusa, which looks like the upside down version of the polyp. And this one is actually mobile. Now, you probably look at this and go, yeah, that looks like a jellyfish. A jellyfish actually does have a polyp form, and they do go back and forth between the two forms. But an animal like a hydra, which you will be looking at under the microscope, has only a polyp form. Sea anemones, I always have trouble saying that word. Sea anemones are similar to corals, and which are also similar to sponges, and that a lot of people think that they're actually plants because they don't go anywhere. But all three of those creatures are actually animals. Now, the interesting thing about the body wall structure of these, you will find that it looks very, very similar to that of the sponge, in that you have two layers with something in the middle. Now, in the book, they do talk about a couple of different terms for it, but it's the same exact type of structure. They talk about an epidermis, which we've seen before, and a gastrodermis on your coloring sheet. But both of those are considered epithelial layers, which is what your book calls them. An epithelial layer just means that you've got a layer of cells on the outside, and then the other side is stuck to something else. And so in both of these cases, the epidermis is kind of stuck to the mesoglea, and so is the gastrodermis. Now, the gastrodermis is on the side of the creature where the gastrovascular cavity is. So this is the first creature that we're going to be talking about that has some sort of a digestive system. And so if you look at a jellyfish, there is a mouth in the center with all these little tentacles hanging down, and they're going to use the tentacles to get food into the mouth and then take it up inside. And then if there are waste products, if there's anything undigested that the creature needs to get rid of, it comes right back out the same way. There is no tube structure like there is in a lot of animals where you have a mouth and you have an anus, and so it goes all the way through the tube and comes out the other end. There's only one opening, and it's both the entrance and the exit for anything that happens to be undigested. Now, if you look at the polyp form, again, very, very similar to that. The gastrodermis is going to be on the inside where the gastrovascular cavity is, and then your epidermis is going to be on the inside with the mesoglea in between the two, and your tentacles up at the top with the mouth up here. The mesoglea is a jelly-like substance, and in jellyfish, that substance that mesoglea is particularly thick, and so that's why jellyfish get their name. In fact, scientists have gone a lot now to just calling them, instead of jellyfish, because they're really not anything like the fish that we know, they tend now just to call them jellies. On the coloring sheet, I did want to point out to you, on the top, you've got the hydra, which is what you'll be looking at under the microscope. On the top right, they call it a physalia, which is the same thing as the Portuguese man-o'-war, similar to a jellyfish. And so that you've got your polyp form and your medusa form. The next row down on the coloring sheet, what they're trying to show you is what the structures look like. If you break the body wall down and look at how everything is put together, I do not know why they left it off, but there really should be on the medusa form. They should also have an I think it was a 7. Yeah, an A7 with an epidermis, because a jellyfish has an epidermis as well. It's not like that layer is missing. Of course, that layer has to be on the opposite side. So you might want to draw that in as well as you're going through that coloring sheet and looking at it. The bottom right corner is actually a cross section of the wall of a hydra, which again, you're going to be looking at under the microscope, and you should be able to see those layers under the microscope when you look at that creature. The really interesting thing about every cnidarian is that they have stinging cells, which are also called nematocysts. And once again, you will get a close-up of this on your coloring sheet to kind of show you how it's put together and how it works, there is always some sort of a trigger on these things. And the triggers are either going to be pressure sensitive, which means if you touch it, or chemically sensitive, which if it comes into contact with some other sort of chemical, it will trigger this. It's almost like there's a lid on this cell, and when that lid pops open, there's this thing up here that has some barbs on it that can bury itself into the prey to help reel that prey in. And then this, it looks like a flagellum, and it's got venom in it, which also is going to sting the prey. You know, it depends on how big the prey is as to what the effect is. If you've ever been stung by a jellyfish, I can't say that I ever have. I understand it's very painful because of the venom that they're injecting. But some creatures are actually going to be paralyzed and even killed by that venom if they're small enough. Okay, so all of your cnidarians are going to have these stinging cells. Whether it's a sea anemone, whether it's coral, they're all going to have that same characteristic. So that's one to really mark down in your mind and say, okay, I know that if it's got a nematocyst, it's got to be in this particular phylum. I just want to look ahead just a little bit, because after we finish this phylum, we're going to be moving into phylum Annelida, which is segmented worms, and we're going to be talking a lot about earthworms. You will not see any videos from me on the earthworm, because instead I'm going to link you to a couple of other videos where I believe they do an excellent job of explaining the structure. And the main reason is because the gentleman that's explaining that structure has a three-dimensional model where he can actually take the parts out and show you how they all fit back in together. So that's why you won't see a video from me on Annelida, but that video is very good. So I encourage you to pay close attention to that as you're studying the earthworm. So kind of took a rabbit trail off to kind of go ahead and explain why you won't see any videos from me on that, but that will also sum up our talk on the cnidarians.